Hey everybody, welcome back to another video. So in this video we're going to talk about the lipoprotein metabolism. We're going to get into big depth here. We're going to talk about how chylomicrons, VLDLs, IDLs, LDLs, and HDLs are formed, how are they metabolized throughout the body, what are their functions, what are their characteristics, and we're also going to talk about the optimal levels for each of these guys in the blood. We're going to talk about their composition and much more. If you guys really enjoy the video, if you learned something from it, truly, please consider liking, please consider subscribing, and let's get right into the video without further ado. So when we first start talking about a chylomicron, a VLDL, and all these other things, the lipoproteins in general, we have to learn a little bit about their general structure, okay? All lipoproteins share an extremely similar structure. What do I mean by this? Well, I mean that they all kind of look like this circular shape, okay? And another thing is that they have specific constituents, okay? And for example, they are all made up of an outside layer of phospholipids, okay? They're all made up of an outside layer of phospholipids. Now, this might look like the same thing that you see in a cell membrane, but it's a little bit different in that the cell membrane is a lipid bilayer, whereas this right here is a phospholipid monolayer, okay? So this is a phospholipid monolayer. And if you guys remember from the structure of a phospholipid, you'll know that it has a polar hydrophilic end and a non-polar hydrophobic tail, okay? So it would make sense that the hydrophilic head is pointing outside to the portion where it will be interacting with the blood or the plasma, right? Because these guys, they have to travel within the blood to reach everywhere in the body. So it makes sense that these little polar heads that are hydrophilic, water-loving, are pointing outwards, and the tails here that are hydrophilic or fat-loving, you can say, are pointing inward. And I'll tell you why they're pointing inward right now. The constituents of the outer portion are the phospholipids and a little bit of free cholesterol. But mainly, what is actually inside the core of this actual lipoprotein molecule is going to be two things. It's going to be these guys right here, a triacylglycerol, and the other thing is going to be a cholesterol, but not any cholesterol, a cholesterol ester. Because remember, a cholesterol ester is just a cholesterol molecule with a fatty acid attached to it right there, okay? So here is your actual cholesterol, but then there's a fatty acid attached to it, which makes it a cholesterol ester, right? Because it's been esterified to it. We talked about this in depth in our previous videos in this playlist. Now, other things that they include on their surface is going to be proteins. But what kind of proteins? These proteins are special. They're called apolipoproteins. Apolipoproteins. And sometimes you can see them abbreviated as APO, or you can see them written as apoproteins. But either way, these apolipoproteins, they are extremely important. And there's two types if you want to get into it. There is a peripheral type, which means they just kind of sit on the outer edge of the membrane. There's an integral one as well, which means it spans the membrane. So this would be a peripheral one, this would be an integral one. Anyways, it doesn't really matter too much. But now you guys kind of know that these are the kind of constituents. So any lipoprotein has an outer layer which is made up of a phospholipid monolayer studded with a little bit of cholesterol. It's got an inner core that's very non-polar, okay, and it's filled with a lot of these triacylglycerols, a lot of cholesterol esters, and much more. We'll look at the composition of each a little bit later, but now you know the general structure, and of course it has these apolipoproteins on top of it. Briefly, what do these apolipoproteins do? Well, these apolipoproteins, you can say they do a couple of things, okay? They give water solubility to these actual lipoproteins. So they give water solubility, and they also regulate key enzymes. Okay, they regulate key enzymes that are involved in lipoprotein metabolism, plus they activate specific receptors. So they act like ligands for their receptor. Activate receptors. So that's pretty cool. That's pretty cool for these apoproteins to do. Okay, knowing a little bit about this is going to help you a lot, a lot. And keep in mind, we're going to talk about different apoproteins, okay? There are many different kinds of apoproteins, and we'll talk about every single one of them. So let's get into it. We have different lipoproteins. The first one is going to be a chylomicron, right here, a chylomicron. This is the biggest one, okay? The biggest in diameter. So what is this? The biggest in diameter, but the least dense, okay? So high diameter, but low density, okay? What about the VLDL? Well, the VLDL, what does that stand for, by the way? Very low density lipoprotein, okay? Very low density lipoprotein. Now, I've put these guys in order of their size and their density. So the further you go down here, all right, the higher the density becomes and the smaller the size. The VLDL, okay, it's got, well, it's smaller than the chylomicron, okay? So I'm going to say smaller than the chylo, but it is more dense than the chylomicron, okay? The IDL and the LDL are interesting molecules. What do I mean by that? So the IDL and LDL are kind of byproducts of metabolism and or extraction of lipids from the VLDL. And you'll see this when I talk about it more in the video, but the VLDL will go through the circulation and it'll get metabolized. It'll get all of these little lipids that are inside of it slowly drained out of him. And the smaller and smaller it becomes, it'll slowly turn into first the IDL, and then second, that IDL will now turn into an LDL molecule, okay, where it's almost devoid of triacylglycerols and only has cholesterol in it. So this is kind of what you're seeing. The VLDL can turn into IDL, and the IDL can then turn into LDL, but they're all coming from this giant VLDL molecule, okay? And I'm not going to go through the rest of this because, you know, the IDL, look at it, smaller than the chylo, more dense. LDL, smaller than the chylomicron, but more dense. The densest and the smallest of all of them is this molecule here, HDL. HDL has the highest density, okay? And it also has the smallest size, okay? What regulates this density, though? Like, how do I know if it's super dense or super not dense? Well, the more lipids that this thing contains, the less dense it is, okay? It's really based upon the lipid to protein ratio, guys. Lipid to protein ratio. If the lipid ratio is higher than the protein, then this lipoprotein is going to be not very dense. But if it has more protein than it has lipids, then it's super dense. And you'll notice once we start going through the percentages that HDL has the most amount of protein, thus it is the most dense. Okay? Now, with all of this said, what are these little numbers that I've put on these apoproteins here, and why do they matter? Well, I'm so glad you asked. Every single lipoprotein is associated with what we call a main apoprotein, okay? A main apoprotein or a characteristic apoprotein, okay? Meaning that apoprotein on the surface of it will help us determine what we're looking at right there, okay? So if I see this ApoB48, I know that I'm looking at a chylomicron. Now this chylomicron might have other apoproteins on it, such as ApoE, ApoC2, and they all serve a function. But ApoB48, that one I can tell. I can tell that with that, this is a chylomicron. What about the VLDL? Well, with the VLDL, I see that it has an ApoB100 and all these other ones. What about an IDL? Well, remember, the IDL is just a byproduct of VLDL, so it will also have B100. Also, what does IDL stand for, by the way? So if this was very low density lipoprotein, this was intermediate density lipoprotein. And then LDL is low density lipoprotein. But anyways, the characteristic difference between an IDL and an LDL with respect to their apoproteins is that the LDL only has ApoB100. Only, okay? That's the only apoprotein that it contains, okay? And that's how you can tell that you're only looking at an LDL there. The HDL is a little different. It has a characteristic protein called ApoA1. Okay, and we'll talk about this guy a little bit more in detail later in the video, but it has the characteristic ApoA1 on it. And that's how you know you're looking at an HDL molecule. Now, with all of this said, what do each of these guys kind of do? Well, a chylomicron is involved in something called exogenous lipid transport. Okay, so the exogenous lipid transport. What the hell does that mean? Well, exogenous lipid transport refers to the fact that this chylomicron is being formed within the enterocytes of the small intestine. So after you've eaten a high fatty food, it is going to take the fats, the fat-soluble vitamins, the cholesterol, and all of that things that was in that fatty diet. It's going to take them all, and inside of that enterocyte, it is going to make this giant molecule called a chylomicron. This chylomicron is carrying exogenous lipids. What does exogenous mean? Not from the body. In other words, it's carrying dietary lipids, all right, to the rest of the body, all right, and then it will end up going to the liver and depositing any remainder there. We'll look at that pathway soon. The VLDL, the IDL, and the LDL, they participate in something called endogenous lipid transport. Okay, so they participate in something called endogenous lipid transport. And this one, again, is pretty self-explanatory. You'll see that the any endogenously made lipids from the liver are going to get packaged into the VLDL molecule, and this VLDL will be secreted from the liver and will be metabolized eventually into an IDL and then an LDL. Okay? And you guys know that LDL gets a bad rep for being the bad cholesterol. Now, why am I putting these on top of it, these quotations? Because there's no such a thing as a bad or good cholesterol. Cholesterol is cholesterol, okay? Too much of a good thing is bad. Too little of a good thing is bad, okay? And I could show you data that proves that too little cholesterol could be just as deadly as too much cholesterol, but that's not the point of this video, so I'm not going to do that. Either way, now you know that these guys participate in endogenous lipid transport. The HDL molecule, it participates in a weird pathway. It's called reverse cholesterol transport. So the reverse cholesterol transport pathway, and we'll talk about this in a little bit more detail at the end of the video, but essentially, you guys should probably already have heard that LDL, what it loves to do is it puts cholesterol into the tissues, okay? That's why it gets the rep of bad cholesterol, because it carries cholesterol and it puts it into the tissues. It deposits it into the tissues. HDL gets the rep of good cholesterol because it's going to take those cholesterol from the tissues and bring it back to the liver, all right? That's why it's called reverse cholesterol transport. It is doing the reverse of LDL. All righty. Now, with all of this kind of in mind, let's take a look at the structural composition of these guys, their lipid profiles. The chylomicron, as you can see, has the most, and this is what I'm going to do. I'm going to circle the most important ones for you. 82% triacylglycerols. Guys, why should this make sense? Well, if you guys remember from the previous video, I mentioned that the majority of the dietary lipids you guys eat is going to be triacylglycerols. Okay, so it makes sense for this guy to have 82% triacylglycerols. It really does make sense. And the rest of it, of course, is going to be the cholesterol, the phospholipids, and the proteins are going to be from the apoproteins. The VLDL, again, mostly triacylglycerol. But remember, these are endogenously made. These are coming from the liver. Okay, it also has some cholesterol in there. Now, we'll dive in depth into where these guys are actually coming from and from which pathways in the liver. Don't you worry. But for now, understand that the most prominent things in this VLDL are going to be cholesterol and triacylglycerols and a little bit of phospholipids. With the IDL, we're not really too concerned here, so I'm just going to skip this, but it is here for you if you want. LDL, though. LDL, what do you think the LDL is going to be high in, guys? I know you know it. It's cholesterol. It's going to be high in cholesterol, and the HDL, well, guess what? It's going to be high in proteins. Okay, why does this make sense? Well, remember I told you that the HDL is the most dense, okay? If it's the most dense, what is the density referring to? It's referring to the lipid to protein ratio, which means what? Which means this guy right here has 50% proteins, and look at the cholesterol in it, 19%, and the rest of the lipids are pretty much minuscule. Okay? So, because of this, it has the highest density out of all of these guys. LDL, of course, super low in triacylglycerols and phospholipids, and very high in cholesterol. Now, I want to explain something to you. Take a notice of the triacylglycerol content in the VLDL, the IDL, and the LDL, and take a look at the cholesterol content of the VLDL, the IDL, and the LDL. What do you notice here? What do you notice? Do you notice how, as we go and metabolize downwards, the content of cholesterol is increasing from VLDL to IDL to LDL, and the content of triacylglycerols is decreasing? That has a reason. That has a reason. If you remember, guys, I said that the VLDL will get metabolized into IDL and then LDL. Well, what is the VLDL super rich in? The triacylglycerols, okay? And so as it goes and circulates, it'll keep getting those triacylglycerols broken down and broken down and broken down, and it'll become super small and turn into IDL. Now the triacylglycerol content is down. Then that IDL will go and do the exact same thing, and it will eventually become so small that it will have like 9% triacylglycerols and turn into LDL. Okay? At that point, the composition of cholesterol relative to the entire size of this particle is now more than it was before. It's 47%. Okay? That's why these two ratios are changing there between the TAG and the cholesterol. Awesome. All right, guys. Now that you know a little bit about the lipoproteins, the different types, the composition of them, and what pathways they're kind of involved in, let's go ahead and dig a little bit deeper into this pathway that we left up on from the last video, okay? If you guys remember, we had those micelles that were created from lipid digestion, and we were going to absorb these guys, okay? And once we did all of that, once we brought them into the enterocyte, we turned all of those molecules into those little small molecules like fatty acids, okay, the monoacylglycerols, and so on. We turned them back into originally what they were: the phospholipid, the triacylglycerol, and the cholesterol ester. Okay, we turned all of those back to what they were, and within the endoplasmic reticulum of the cell, this enterocyte, we added some proteins on the surface of it, like ApoA, like ApoB48, and then we put this guy out into the circulation. What did we call that guy? Oh, and also, we also put the fat-soluble vitamins in there. What did we call this guy? We called that guy a chylomicron. This chylomicron will then drain away from this enterocyte, not into the blood, but first into a specialized lymphatic vessel called a lacteal. Okay? Again, we talked about this in a previous video, but it'll drain into that lacteal, and then from the lacteal eventually drain into the thoracic duct, which is now the actual circulation. So now we're going to look at what happens to this chylomicron as it's making its way into the circulation, and actually as it is in the circulation. All right, guys. So a little bit more about this chylomicron now. Before we actually go ahead and draw the pathway, let me actually tell you something. Let me tell you something. This ApoB48 that's on top of it, right? This was made inside of the small intestine along with this other apoprotein that's not shown here called ApoA1, okay? And the B48 and the ApoA1 are going to be present on the chylomicron as it is being secreted outside of the enterocyte, okay? Now this ApoB48, a little bit more about it is, take a notice how it's the same color as this ApoB100, okay? The only difference between these two guys, it's something very funny actually, is that the ApoB48 is made by the intestines and the ApoB100 is made by the liver. But funnily enough, they're actually coming from the exact same gene, okay? However, in the intestinal cells, the enterocytes, they actually contain a stop codon that stops the protein at 48% of the shape of ApoB100. That's why it's called ApoB48, okay? But anyways, that's what I wanted to tell you. I wanted to tell you that this ApoB48 is coming from the exact same gene as the ApoB100. However, it's just because that it has a stop codon prematurely, it makes it 48% the size of the other one. All right. Now, as this actual, and let's draw it out here. Okay. As this actual chylomicron right here is going to be entering the circulation, what is it going to have on it, guys? It's going to have ApoB48 and ApoA1. But guess what? Guess what? There is another molecule in circulation at this current moment in time. And this molecule is called HDL. This HDL is so kind. You know what he's going to do? He's going to come here and he is going to say, hey, you know what, chylomicron, you don't have enough proteins on your surface. Okay? You don't have enough proteins on your surface. I'm going to give you some extra proteins. And those extra proteins are going to be ApoC2 and ApoE. Okay? So now what you're going to notice is that this chylomicron will now have two extra proteins on its surface. Okay? It'll have an ApoC2, it'll have an ApoE, an ApoB48, and an ApoA1. Okay? Awesome. And who gave him that? HDL. The HDL molecule gave him that. Now, why is it important? Now, you got to remember, this chylomicron, what is it super rich in? It's rich in triacylglycerols and mainly cholesterol. Okay? So let's leave that there. Triacylglycerols and mainly cholesterol. Another thing you have to know is that these chylomicrons, guys, they're super slow. They enter the bloodstream really slow. Why? Because they have to go through the lacteal first, that lymphatic vessel. And so it takes them a long time to actually get inside of your actual bloodstream. Well, how long, you may ask? Well, usually you might even see these guys enter the blood for up to 14 hours after the consumption of a meal. Okay, of course these numbers can differ depending on how fatty the meal was and the stomach digestion time and all that stuff. But this is why that when you usually go and get a blood lipid test, they say that, oh, I want you to have fasted for at least 12 hours so that they can get a true reading of your blood triacylglycerol levels and all that stuff that are actually devoid of chylomicrons from your previous meals. Okay, so what happens now after this HDL gives this guy, this chylomicron, this ApoC2 and ApoE? Well, this chylomicron will continue circulating until it comes across certain tissues. What kind of tissues? These tissues could be adipose tissue, the heart, or the skeletal myocytes. And in each of these tissues, okay, the capillary beds contain a specific enzyme called lipoprotein lipase. Read the name. So lipoprotein, okay, so we know chylomicron is a lipoprotein, and then lipase. If you guys remember from the digestion video, we said a lipase is anything that is able to cut a triacylglycerol, okay? And so what is this guy going to do? This lipoprotein lipase will act on this chylomicron and cleave all of its triacylglycerols, okay? But how? How does this enzyme here become activated? I'll tell you how this enzyme becomes activated. This lipoprotein lipase, okay, it becomes activated by ApoC2 right here. So this friendly guy HDL gave us this ApoC2 that we were lacking, but now that we have it, we can use this ApoC2 to activate the lipoprotein lipase, where then it's going to cleave the triacylglycerols inside of this guy, okay, and release specific molecules. What kind of molecules? Free fatty acids and some monoacylglycerols. All right, these guys can be used by these tissues for energy production and storage. If we're talking about the heart and the skeletal muscle, they're most likely going to be used for energy production. If we're talking about the adipose tissue will use these free fatty acids, these monoacylglycerols, to make triacylglycerols and store them, okay, as fat. Now you know what this chylomicron is doing, okay? It is bringing exogenous or dietary fat to tissues for use. How is it doing that? With the help of this lipoprotein lipase that's being activated by ApoC2, cleaving this guy and allowing for that fat to become free. Okay, that's awesome. But you know we're missing something? Triacylglycerols are made up of what? fatty acids and a glycerol molecule. Well, we release the fatty acids and the monosaccharides, but what about the free glycerol? I'll tell you, there is actually a free glycerol circulating here now. Okay, here's that free glycerol, and I'll tell you where this free glycerol goes in a second. But first, what happens to this chylomicron? Well, as this chylomicron circulates and keeps getting cleaved by the lipoprotein lipase, eventually it'll become super small, and it'll turn into this thing right here. Right, you see how it's smaller? And that thing, it still has some levels of triacylglycerols and some levels of cholesterol, but not anywhere closest to the original amount. This guy is now called a chylomicron remnant. So a chylomicron remnant. I'm going to abbreviate it here as CR, and this one was just chylomicron. This one is a chylomicron remnant now. You know what this chylomicron remnant does? This chylomicron remnant comes back to this HDL, okay, and say, hey, you know what, HDL? Thank you so much for renting me those proteins that you gave me, okay? I'm going to give you back some of them. I'm going to give you back A1 and ApoC2. But you know what it keeps with it? It keeps the ApoB48 that it originally had on, and it keeps the ApoE that it got from the HDL, okay? So you notice at the start, the HDL gives it ApoC2 and ApoE, but then when the chylomicron remnant gives it back, it only gives it back the ApoA1 and the ApoC2, but it keeps the ApoE, okay? Now, this chylomicron remnant here will now continue to circulate until it reaches the actual liver, okay? So it goes to the portal vein, and then it reaches the liver right here. So let's show that. Now it's at the liver. Here we go. It's at the liver, and again, it's got some triacylglycerols, it's got some cholesterol in it, and we got the ApoE on it, and the ApoB48. Once it makes it to the liver, it is going to interact with a very special receptor, okay? This special receptor actually has a name. It's called LRP1, okay? It stands for the LDL receptor related protein 1, okay? So LDL receptor related protein type 1. And what is going to happen is that this ApoE, so one more time, this LRP1 recognizes the ApoE on the surface of this chylomicron remnant, okay, and it allows it to be taken in to this actual liver cell. What is a liver cell called? It's called a hepatocyte. So it allows it to be coming inside of this actual liver cell, and at the same time, this enzyme that is located very closely to this receptor called hepatic lipase, or HL, okay, hepatic lipase, will, look at it. Here's the chylomicron coming in. It'll bring in some cholesterol with it that it had in it, and it'll bring in some triacylglycerols. But guess what? Those triacylglycerols that it's bringing in will be acted on by the hepatic lipase to create some free fatty acids and glycerol. Okay? So that's what's the fate or the end of the story for the chylomicron remnant. Okay? The chylomicron started off circulating like this, okay? And then it became super small and then turned to a chylomicron remnant. Now, another additional note that I'll give you guys is that how much of the triacylglycerols are cleaved by the liver and how much of the triacylglycerols are going to the tissues. Okay, well, about 80% of the triacylglycerols that are inside of the chylomicron will go into the tissues being acted on by the lipoprotein lipase. And the remaining 20% will be acted on by the hepatic lipase whilst being taken inside of the hepatocyte or the liver. Okay? Now, that doesn't finish our story just yet, because if you remember, at this level, we made this glycerol, and I said I'd tell you what happens with this glycerol in the liver. Good thing you ask. Well, this glycerol in the liver, okay, not the liver, sorry, in the circulation, can go to the liver. Once it goes to the liver can use this glycerol and this one that we actually made here, okay? It can use it to go through a process called gluconeogenesis. Gluconeogenesis is a process in which we use non-carbohydrate sources such as amino acids, such as lactates, such as glycerol, to make glucose, okay? And once we make this glucose, we can do a couple of things from it, okay? That glucose from the gluconeogenesis can be turned into glycogen, okay? Or it can be turned back into glucose from glycogen. Now, you guys remember that turning glucose into glycogen was called glycogenesis, And breaking down glycogen to glucose was called glycogenolysis. Either way, you have this glucose. Now, what ends up happening is sometimes in the liver, after eating, you have an abundance of glucose. So much so that this glucose can be shuttled into a different pathway. What kind of pathway? Well, this glucose can be shuttled into making a molecule called malonyl-CoA, which you don't have to worry about too much. But you have to know that this malonyl-CoA can be used, actually, to make a free fatty acid. Okay? So you see what the liver just did? The excess glucose that it made was turned into a fatty acid. Okay? It was turned into a fatty acid. Now, what else can this liver do? What else can this liver do? Well, it can do the exact same thing with this glycerol that it got from the chylomicron remnant as well. But do you guys remember that we had another pathway? We had another pathway in the liver that was called endogenous lipid transport. And when we start accumulating a lot of lipids inside of the hepatocyte, like the free fatty acids from this pathway, like the cholesterol and the fatty acids that we got from the chylomicron remnant, the liver starts taking those guys, okay? So look at it. It's going to take this guy, it's going to take this guy, and it's going to take this free fatty acid from here, okay? And it's going to package it, okay? It's going to package all of that into this molecule here called a VLDL, okay? It's going to package it into this VLDL molecule, all right, inside of the endoplasmic reticulum, and then push it out into the Golgi apparatus and you know, modify it a little bit there. But this molecule will contain ApoB100 on it. It'll contain ApoE, and if you want, technically it will also have ApoC1 on it. But you know what? I'm just not going to put that. That will confuse some people. Okay? So it will technically have ApoC1 on it as well. Anyways, so what is this guy going to have? It is going to have triacylglycerols. It's going to have cholesterol, and we know, of course, it's going to have other things like phospholipids and stuff. But we know that the main things that it's high in are going to be triacylglycerides and cholesterol. Now, notice something. This is the endogenous lipid transport pathway. So that means that anything that the liver endogenously makes is going to package into these. But wait a minute. I just told you that it's taking the fatty acids and the cholesterol from the chylomicron remnant as well. Yeah, exactly. It can do that as well. Okay? So another thing that it can do is, you know, the liver has its own storage of triacylglycerols. Okay? So hepatic triacylglycerols can also be incorporated within this actual VLDL if necessary. Another thing, you know, the liver is one of the many organs that can abundantly make cholesterol. And why can it do that, by the way? It can do that because it needs the cholesterol to make bile, guys. Okay, you guys should remember that. All right, but let's go ahead and really quickly walk through the pathway real quick. So you can make cholesterol from this precursor molecule called acetyl-CoA. But where do you get acetyl-CoA? Well, you can get acetyl-CoA from many different sources. You can get it from amino acids, you can get it from fatty acids, you can get it from glucose, okay? But once you get this acetyl-CoA, you can push it into a cholesterol synthesis pathway if necessary. And this is actually going to be extremely brief because there's 21 steps to cholesterol synthesis, and there's no way I'm going through all of that. But acetyl-CoA can be turned into HMG-CoA, all right? And the HMG-CoA, through the help of a specific enzyme, can be turned into mevalonate, and the mevalonate can be turned into squalene, and the squalene can eventually be turned into cholesterol. Okay? This cholesterol that I made right here, it can also be incorporated into that VLDL molecule. Okay? So that's from the endogenous pathway. Now I told you that there's a special enzyme here that's converting HMG-CoA to mevalonate. That's actually called HMG-CoA reductase enzyme, okay? And actually, people who are taking cholesterol-lowering drugs like statins, okay? Statins, those drugs, statins, like Lipitor, for example, they target this HMG-CoA reductase, and then they inhibit this enzyme so you don't make cholesterol, so you can lower your cholesterol levels, okay? However, that has had limited success. And the reason why they target this enzyme is because this is the rate-limiting step. If any of you have taken biochemistry, you'll know what that means. Rate-limiting enzyme is the enzyme that if you block it, the rest of the pathway cannot move on. It can back up. What is the whole point of all of this that I was trying to tell you? Well, the whole point is that from the liver's metabolism, okay, we made cholesterol, and we can incorporate that cholesterol into the VLDL, okay? From the liver metabolism, we also made fatty acids from scratch, okay? That's de novo lipid synthesis. The liver also has its own storage of triacylglycerols that it can incorporate into this VLDL. But the liver also got these chylomicron remnants that had cholesterol and free fatty acids. But guess what? We don't want to accumulate too much fat in the liver because that can lead to hepatotoxicity. So what we do is we push it out all into this VLDL, and this VLDL will then be pushed out into the circulation. Last thing I'll say about the cholesterol that we make in the liver is that it can be incorporated to many different areas. So like I said, it can be incorporated into bile salts, which will then be put into the bile, okay? Or it can just be freely put out into the bile. Another thing is that it can actually be incorporated into the cell membranes of the actual hepatocyte, because you guys know that cholesterol can participate in membrane fluidity and all that stuff. So that's a little bit on that. Now let's finally follow this VLDL molecule. So like I told you, this VLDL molecule, it's got ApoB100, ApoE on it, and it was made in the endoplasmic reticulum, then in the Golgi, and then pushed out, right, exocytosed out of this actual hepatocyte into the circulation. So now let's follow this. So here we go. Let's push this guy out into the bloodstream, okay, and see what it's going to do. And again, remember that the main apoprotein on the surface of this VLDL is ApoB100, and in fact, there is one molecule of ApoB100 per each VLDL molecule. So here he is. Here's that VLDL. And you know what? We got to make it a little bit smaller so we can have space moving forward. So here's the VLDL, guys. It's got the B100 on it. It's got the ApoE on it. And then we have this good old friend that we have back here. You guys remember this good old friend we had back here? HDL. HDL comes up to this guy, to this VLDL molecule, and says, you know what, buddy? You don't have some proteins, okay? I'm going to give you those proteins. Why am I going to give them to you? Because I like you, man. So this HDL will come here, okay? And it'll go up to this actual VLDL and say, hey, you know what, buddy? You're missing some key proteins, okay? I see you already got some. It says, hey, man, I know you already got some ApoE, but I'm going to give you some more ApoE. And on top of that, I'm going to give you ApoC2, okay? So look at it. It's going to give him some ApoC2, and it's going to give him some ApoE. And now what is it going to look like? Now look at this guy. Okay, this funky guy over here, he's going to come here, and he is going to look like an absolute boss, okay? Look at him. He's going to have some ApoB100 on it. He's going to have some ApoC2 on him, and now he's going to have some ApoE, more ApoE as well, okay? And you guys remember that this guy was primarily consisting of triacylglycerols and cholesterol, okay? And so what's going to happen? This guy right here, the entire purpose of making this VLDL, right, was pushing the endogenous lipids that come from the chylomicron remnant and the metabolism of the liver out from the liver and pushing them to the peripheral tissues when we need them. Now, something you should note, by the way, is that the chylomicron pathway, the exogenous lipid transport, that system is only operating after a fat-containing meal, okay? And then they disappear after all the dietary triacylglycerols are delivered to the target tissues, okay? But the endogenous lipid transport system, it operates continuously. It operates continuously. Why? To maintain a proper balance of fatty acids and triacylglycerols accumulating in the liver during just normal metabolism, okay? So this pathway is occurring continuously, whereas the chylomicron pathway is not. It's only occurring after a high-fat meal, or just, fat-containing meal. Now, what happens to this VLDL as it comes close to this tissues? Now, you remember what these tissues expressed? They expressed the lipoprotein lipase, guys. What activated that? The ApoC2. So look, the ApoC2 is going to activate the lipoprotein lipase, and what's going to happen? The lipoprotein lipase will keep getting cleaved, all right, and get those triacylglycerols out of there. What happens to those triacylglycerols, guys? Guess what? They will get broken down into free fatty acids and some monoacylglycerols. And of course, a free glycerol molecule, which guess what? It can go to the liver and go through gluconeogenesis. Awesome. Okay, this becomes the tricky part. What happens to this VLDL molecule after it has become cleaved a certain amount? Well, here's the thing. This guy can turn into an intermediate molecule. It can turn into this intermediate molecule here that we talked about. It's a bit smaller, actually, and less and more dense. This guy is called an IDL, or an intermediate density lipoprotein, and it still contains ApoB100, but now you'll notice that it only has ApoE on it. What happened to the C2? I'll tell you. As this VLDL was being cleaved and turning into this IDL, it ended up returning to HDL the ApoC2 that it took from it. Okay? Now, this IDL molecule that we made, what good is this to us? What can we do with this IDL molecule? I'll tell you what we can do with this IDL molecule. This IDL molecule, it can either stay at this lipoprotein lipase site and keep getting cleaved. Again, what will it have in it? It'll still have some triacylglycerols, but now it'll have a little bit more cholesterol than before. Okay? So it can keep staying at that lipoprotein lipase site, and I'll show it like this. Okay? It can keep staying there, where the lipoprotein lipase will keep acting on it, and eventually it'll keep acting on it so much, cleaving all those triglycerides so much until it's almost devoid or empty of triacylglycerols, and it turns into this other molecule. What's that other molecule, guys? It's this very, 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 very small molecule called an LDL molecule, which only has cholesterol in it now. Okay? And guess what? It only has ApoB100 on it. Okay? Which means what happened to that ApoE? That ApoE must have been given back. So in this process where the IDL decided to stay here and get cleaved more and more by the lipoprotein lipase and turn into this LDL molecule, right then and there, as it's turning, it is also going to give back the ApoE to the HDL molecule. But you know, none of that could happen at all. It could also, this IDL molecule, it could also just say, you know what, screw all this, I'm out. I'm going to go to the liver myself. And what it can do with the liver then is interact with this LDL receptor. Okay, and you're probably thinking like, wait, hold on a minute. How does the IDL molecule interact with the LDL receptor? I'll tell you how. Remember the IDL, it has this ApoE on it? Well, this ApoE molecule, it can interact with the LDL receptor. What else could it interact to? You guys remember it can interact with the LDL receptor related protein 1? Exactly. So the ApoE can not only interact with the LRP1, but it can also interact with the LDL receptor. And if it does that, guys, what's going to happen? This IDL that will come here and interact with this, it will get endocytosed in, okay, and it'll get broken down and release some cholesterol and some free fatty acids, okay? Nice. And you know how it gets broken down, right? It'll get endocytosed, and it'll get combined with the lysosome, and inside of the lysosome, it'll get broken down into cholesterol and free fatty acids. Awesome. So two fates for this IDL. It could either go to the liver and interact with the LDL receptor, all right, and get taken up and destroyed, or it can remain at that LPL site, keep getting cleaved, and turn into an LDL. Now, this is the more likely pathway. This guy will turn into an LDL molecule, all right, and lose that ApoE, giving it back to HDL, and remain as this ApoB100 LDL molecule now. What is this LDL going to do? The thing that you guys all know it is going to do. It is going to transfer cholesterol into the tissues. How? Look at it. It's going to come to peripheral tissues, non-hepatic, by the way. Non-hepatic tissues, okay? It can do the same thing in the hepatic tissues, but non-hepatically. It'll bind onto its receptors, okay? And it'll cause the deposition of your good old friend cholesterol, okay? So it'll cause the deposition of cholesterol inside of these actual Non-hepatic tissues. So what is this? This is a non-hepatic tissue. What kind of non-hepatic tissues, guys? Well, this could be in many places, okay? Many places that need cholesterol. For example, there are steroidogenic places like the testes, the ovaries, the adrenal glands. They can use this cholesterol to make steroid hormones, okay? Or it could just go to the liver, and in the liver, once it deposits that cholesterol, you guys know, and I'll put an arrow here, it can come to the liver. And then once it comes to the liver, guess what? It'll also release some cholesterol, which we know what we can do with that cholesterol, right? We can put it as a part of the membrane. We can put it into the bile. We can turn it into bile salts, then put it into bile, right? All that stuff. Or, unfortunately, this LDL molecule might contribute to the formation of an atherosclerotic plaque. Now, I'm not going to get too deep into it, but it has been indicated that large buoyant LDL molecules, okay, that are not oxidized are pretty much benign LDL, and that small and more dense LDL molecules seem to be more easily oxidized, and they seem to be the problem in atherosclerotic plaques, okay? That's why there's a push to not only test for LDL molecules, but look at the subfraction of what kind of LDL molecule am I actually looking at in the blood of this patient, okay? But anyways, that's that. Now, how much of the actual LDL is going to be removed by the non-hepatic tissues and how much by the actual liver? Well, I'll tell you something right now. The liver, okay, is extremely important. The LDL receptors located on the hepatocytes are really important. Why? Because these guys, they remove about 70 to 80%, 70 to 80% of LDL particles from the circulation, okay, from circulation. So the amount of LDL receptors on the actual liver really matters, okay? And you'll see that you can actually change this with your diet. What do I mean by this? Well, for example, the consumption of saturated and trans fat has been known to decrease the number of LDL receptors, which might have an impact on your circulating cholesterol level, right? That means if there's less LDL receptors, we're not taking up this much, and therefore you're going to have more LDL cholesterol in your circulation, which is not good, because the more you have, the higher the chance that it might get oxidized and then cause an atherosclerotic plaque. On the other hand, phytosterols and soluble fibers have been known to increase the number of LDL receptors on the liver, meaning more clearance of LDL, meaning less chance of developing atherosclerosis and cardiovascular disease. Either way, that's that. Now, very briefly, guys, let's take a look at the interaction that occurs between the LDL molecule and the receptor, okay? So I've gone ahead and made this image here for you guys. And now over here, you can see the normal LDL and LDL receptor interaction, okay? So first things first, the LDL particle will bind to the LDL receptor here in blue, okay? And here's the LDL particle. After it does that, it will get endocytosed inwards, okay? Where you'll have this endosome containing both the LDL receptor and the particle. But then something cool happens. The LDL receptor, okay, will then become separated from the LDL particle, and the LDL receptor will become recycled and go back to the surface of this actual cell, whereas the lipoprotein itself, the LDL, it will go and fuse with a lysosome and become degraded and release all of those, cholesterols and all that stuff that was in it, okay? So that's the kind of pathway. And in fact, something cool is that the lifespan of this LDL receptor is about 20 hours, okay? And within that lifespan, it will go in and out of the cell. It'll recycle in and out of the cell about every 10 minutes, okay? So every 10 minutes, it'll keep doing this, and it has a lifespan of 20 hours. That's pretty funny. Now, something can actually change this normal LDL-LDL receptor interaction. There is a molecule called PCSK9. Now, this PCSK9 stands for a giant name. It's called proprotein convertase subtosylin kexin 9. I think it's just better to suggest that it's PCSK9. But it is a secreted protein. It's mostly coming from the liver. However, it can be found a little bit in the small intestines and the kidneys. But what it does is it is going to be secreted and it will bind to the actual LDL receptor and cause the intracellular degradation of the intracellular receptor of the LDL receptor. Okay, so how does that look like? Well, let's take a look at it. So here in green is the PCSK9 being secreted. Here's the LDL, and then blue is the LDL receptor. Okay, so what you're going to notice here is that this PCSK9 has already been secreted and it's already bound to the LDL receptor. It's technically bound to it in an open conformation. What's going to happen is now this guy's going to get endocytosed, okay? It'll go and turn into this endosome, and because of the presence of this LDL receptor bound to the PCSK9, it will no longer be recycled, but rather it'll go and fuse with a lysosome, and the LDL receptor, the LDL, and the PCSK9 will all become degraded. They will all become degraded. So you can see how this right here, this PCSK9 molecule, has a big impact on the number of LDL receptors present on the surface of the cell. What kind of cell? Well, remember, I told you that it's mostly secreted from hepatocytes. Okay, and so it would make sense that this PCSK9 regulating the actual LDL receptor number on the hepatocytes may have a role in regulating blood cholesterol levels. So what people have done is they started to take an advantage of this, and they have developed PCSK9 inhibitors to manage LDL cholesterol, okay? And it can actually lower LDL cholesterol pretty good, pretty decently. In fact, PCSK9 inhibition has been shown to cause an additional 50 to 70% decrease in the LDL levels in individuals who are already taking statins. Okay, so in conjunction with the statin, they took a PCSK9 inhibitor and they saw a 50 to 70% decrease in the LDL levels. That's pretty impressive. So with all of that said, okay, you guys now know how the chylomicrons are working, how the VLDLs are made, and how they're turning into an LDL molecule. What about this pronounced HDL? What about that guy? This HDL has been helping us all steps of the way. It's been, giving apoproteins to us. It's been giving apoproteins here. What does its pathway look like? We said it's called the reverse cholesterol pathway, but what does that actually look like? Now, guys, there is a complex version of this, and I'm going to give you the non-complex version here first, but if you're interested, you can go and I'll put chapters in this video so you can skip to the part that has the more complex version, okay? But I'll first start with the non-complex version. Okay, so starting off, this actual HDL molecule can come from two places, okay? It can come from the small intestine, and it can come from the liver, okay? And it's going to start out in a discoidal shape. Quite literally, it'll have this discoidal shape to it. And we know that it has mostly proteins, but the other things that are pretty high in it are going to be phospholipids and some cholesterol. It also has another thing in it. It's got a very special enzyme inside of it. That enzyme, guys, it has a name. It's called, and let's draw it here, it's called LCAT. Okay? The LCAT stands for lecithin cholesterol acetyltransferase. But remember, I also said that it has, it's pretty abundant in proteins. So what kind of proteins does it have? If you remember from the start of the video, the characteristic protein it has is called ApoA1. It has some other proteins such as ApoC2 and ApoE. Now, like I said, it can be made from the small intestine and the liver, but more so the liver than the small intestine, okay? Now, what is this guy going to do? So first of all, it starts out in this kind of nascent HDL form. It's called nascent, okay, or discoidal, whatever you want. Nascent or discoidal shape because it looks like a disc. And it will circulate, okay, it'll circulate towards this non-hepatic tissue, okay? What kind of non-hepatic tissue, by the way? It could be the adipose tissue. It could be the muscle. It could be an atherosclerotic plaque, okay? In the arterial wall, okay? So any place that has cholesterol deposited in it, it'll go there. And let's draw what it is, okay? So here it is again. Here's that actual nascent or discoidal shaped HDL molecule, okay? And what does it have on it? It's got the ApoC2 on it, it's got the ApoE, and it's got the ApoA1. It's got some phospholipids in here, okay? It's got some cholesterol, and it's got that famous enzyme, LCAT. We'll talk about this. So what's going to happen is that this ApoA1 is going to interact with this specific receptor on this tissue. Okay, this specific receptor on this tissue is called ABCA1, or the ATP binding cassette transporter A1. Why A1? Because it's associating with ApoA1. Okay, so this guy is going to interact with them, and it's going to cause the uptake of cholesterol from this tissue into the actual HDL molecule. And this HDL molecule, once it takes up this cholesterol, with the help of this specific enzyme, lecithin cholesterol acetyltransferase, it will turn this cholesterol into a cholesterol ester. If you guys remember from the digestion video, we had another enzyme inside of the endoplasmic reticulum that was called ACAT, acetylcholesterol acetyltransferase. It's very similar in function. So over here, what this guy is doing, this LCAT, is, look at its name, lecithin. Again, if you remember from a previous video, lecithin is a phospholipid. So it's taking this phospholipid, lecithin, okay, and it's cleaving a fatty acid from it to create a lysolecithin. Then it's transferring the actual free fatty acid from that phospholipid that we just cut onto the cholesterol, okay? So that's called lecithin cholesterol acyl. Acyl is a fatty acid transferase, okay? So it's putting that fatty acid from the phospholipid that we just chopped off into the cholesterol to make a cholesterol ester. Why is it doing this? Well, it's doing this, one, to change the shape of this actual HDL molecule from a discoidal shape to a more circular shape, okay? And we'll look at that in a second. But it's also doing it to allow diffusion of more cholesterol inside, okay? Alrighty, so now let's look at it. Now let's see what this cholesterol, this HDL molecule will look like after it has gone through this cholesterol uptake. Now it'll have a big old belly. Look at it. It'll have a big old belly, and it's going to look like this. It's looking all circular and big now. Now it's got phospholipids in it, it's got cholesterol in it, and it's got cholesterol esters in it. Okay? And it still has that LCAT enzyme. Okay, it's got that LCAT enzyme in it. But now it's no longer a nascent discoidal, it's now a mature spherical HDL molecule, okay? This HDL molecule, it has two paths. It's two paths. And its main path for now is going to be that it will return back to this, okay? So look at this. I'll do it like this. It can go to the liver, okay? And then I'll put an arrow back here. And I'll tell you why I did that. So it can go to the liver and interact with a specific receptor in the liver called SRB1, or scavenger receptor type B1, okay? Once it interacts with this guy, it'll cause the release of cholesterol, or the deposition, I should say, of cholesterol into the liver, and then it will continue to circulate in this discoidal shape. Why? Well, because it deposited all of that cholesterol and cholesterol esters, if you want, into the liver, emptied itself out, became discoidal and nascent, and continues throughout this HDL cycle, okay? After it has done its cycle, all right, like finally done all of its cycles, it will come back to the liver, interact with that same SRB1 receptor, and it will now get endocytosed, releasing its cholesterols and also releasing some fatty acids, okay, because it had those phospholipids in there. And then it'll finally now be done its life cycle. So what is its life cycle? Okay, well, the HDL molecule, guys, it has a lifespan of 2 days, okay? I know, it's shocking. Lifespan of 2 days. In those 2 days, each particle of HDL does about 7 cycles, okay? So 7 cycles each HDL. So this guy will do 7 cycles before he comes to the liver and then gets degraded. Okay? Now, another thing to mention about the HDL is that it has certain interactions with other lipoproteins in the blood. And you guys have already seen some of those interactions, okay? You've seen that it can donate some apoproteins to them, but it doesn't only do that, okay? It does other things as well. It has this enzyme called cholesterol ester transfer protein and this other one called phospholipid transfer protein, where it'll go and interact with VLDLs and perhaps an LDL molecule as well. That has been shown. And it'll transfer some of its cholesterol esters to these actual VLDL molecules, and instead it'll pick up phospholipids, okay, from them. What does this do? Well, the HDL molecule will transfer its cholesterol esters because it helps the HDL to reduce its size so that it has an easier time interacting with the cell surface receptors to pick up more cholesterol now, okay? And in fact, it picks up more phospholipids as well. It has been shown to, or perhaps it's been said that it might do this as well with chylomicrons, but I'm not going to include it here because it's not big evidence for it. But it has been very well known to do this with VLDL and LDL. Now, finally, just to talk briefly, guys, just to talk very, very briefly about the optimal levels for all of these guys, and then we'll finish off the video with the actual complex version of the HDL cycle. Now, total cholesterol, the desirable amount, and this, by the way, these are the most recent amounts. The desirable amount for total cholesterol should be less than 200 milligrams per deciliter. Deciliter is just 100 milliliters of blood. If it's too high, it's going to be greater than or equal to 240. HDL cholesterol, you want this guy to be as high as possible. So a desirable amount is greater than or equal to 60. It's very risky if it's less than 40 milligrams per deciliter, okay? With the LDL cholesterol, you want this guy as low as possible, okay? So optimal level is less than 100 milligrams, but there's values even as low as less than 70 that have been seen to be pretty good. High level is anywhere between 160 to 189. Like very high is going to be like greater than 190 milligrams per deciliter, okay? Normal levels of triacylglycerols in the blood are going to be less than 150 milligrams per deciliter. And of course, a high level would be something around 200 to 499. Okay, that's insane levels. Again, milligrams per deciliter. Alrighty, so now let's go ahead and talk about the actual complex version of the HDL cycle, okay? Because that one is like the one that is super complex, and for students who are not that advanced, I don't want to go into too detail. For those of you who are done watching the video, like I really appreciate it if you stuck around this far. I highly recommend you go and watch the previous videos in this playlist because it just ties in with everything we've been talking about so far. And I would really appreciate it if you guys like the video, if you subscribe, maybe leave a comment. It helps with the algorithm. And yeah, let's finish up this video with this HDL cycle. So here is the actual real pathway for the HDL molecule. So unlike chylomicrons and VLDLs, which we saw that were actually made and assembled into a complete lipoprotein inside of the cell, that doesn't actually occur with the HDLs. They completely arise inside of the actual intravascular space, right, in the blood vessels. So, okay, what do I mean by the fact that it's assembled completely inside of the intravascular space? So, remember how I said that it can be coming from the intestines and the liver? Well, this is what I actually mean. So, the intestines and the liver, they secrete the apoprotein ApoA1. So they secrete ApoA1, right? And they literally just put out this protein into the circulation. This ApoA1 will circulate. It'll circulate here, okay? And this lipid free, because this guy is currently lipid free, okay? It has no lipids on it. It's just the ApoA protein. But this lipid free ApoA1 is secreted from the intestines and the liver. Now, if you want, it can also technically be secreted from VLDLs and chylomicrons when they are going through triacylglycerol hydrolysis, okay? But the whole point is this ApoA1 makes it into the blood. And the way this ApoA1 turns into a lipoprotein is that it first has to become lipidated. What does that mean? It has to acquire some lipids. What kind of lipids? Phospholipids, some cholesterol, all of that stuff. So how does it do that? Well, this ApoA1, it acquires the phospholipids and cholesterol by interacting with the actual liver's ABC A1, the ATP binding cassette A1. Now, if you remember, in the simplified version, I did say that technically the ABC A1 is interacting with the ApoA1 and allowing for the movement of the cholesterol and phospholipids and stuff outside of that actual cell. So that's what's happening here. After this ApoA1 interacts with this receptor, it acquires some phospholipids and some cholesterol. Here, you can see that. Those phospholipids, those cholesterol, they're going to go through here, and they're going to give rise into what? They're going to give rise to this little nascent discoidal-shaped HDL that we know. Okay? So that's how we're going to get that nascent discoidal HDL, guys. It will now, on its surface, have that ApoA1, okay? And inside of it, it will now have a little bit of that phospholipid and a little bit of that cholesterol, okay? Now, so look at what this guy's going to do now. Look at him. He's going to come here, and he's going to interact with this new kind of scenario here. It's going to come here, and it's going to interact with the non-hepatic tissues now, all right? And it's going to interact with two receptors, actually. It's going to interact with the ABCA1 and the scavenger receptor beta 1. So what do I mean by this? Look at it. Look at this APO. And you know, let me erase this here, make it all nice. ApoA1 here. And this guy will then come here as the discoidal shaped. Look at him. He's got some phospholipids. He's got some cholesterol, okay? And now guess what? He's going to get even more phospholipids and cholesterol from these guys, okay? And turn into an even bigger version of the discoidal, okay? He's still discoidal, though. And he's still got that ApoA on it, okay? ApoA1, okay? But as he accumulates all of these guys, okay? As he accumulates all of these, he's no longer going to be a discoidal-shaped guy. You know what's going to happen to this guy? Look at him. As he's going to circulate, a couple of things are going to happen to this HDL molecule. It will now turn into a mature HDL molecule, okay? So it will now become mature. And on the way, it'll pick up something, okay? It'll pick up an enzyme that is just floating around in this intravascular space. This enzyme is called LCAT. So it'll pick it up and it'll now reside within itself, okay? Now that it does this, guess what it's going to do? It will keep circulating now, okay, and come into some more non-hepatic tissues. And over here, it'll interact with these two receptors again, but something's different. Because it's a mature LDL molecule, it will no longer interact with the ABCA1. It will now interact with the ABCG1. Okay, a mature HDL can never interact with the A1 again. However, it can interact with the ABCG1 and the scavenger receptor B1, no problem. Okay, so look at it. It'll take cholesterol again from here and from here. Okay, it'll incorporate it into itself. And with the help of this lecithin cholesterol acetyltransferase, we are going to make cholesterol esters. Now, one more time, why does this actually occur? So, like, how does this actually occur? Sorry about that. So this occurs like this. You guys remember we had this enzyme here called phospholipase A2, and we had this lecithin here, and the phospholipase A2 was coming here on the SN2 carbon, and cutting this actual fatty acid that was attached to the SN2 of the phospholipid. That's exactly what this LCAT enzyme is doing. The LCAT enzyme, okay, it's coming right here at that SN2 position. It is going to cut there, and then it's going to take this fatty acid and transfer it to this cholesterol molecule, okay, and turn it into a cholesterol ester. That's literally all it's doing, okay? And we know that there was already phospholipids inside of this actual HDL, okay? So that's what's going to happen here. And this HDL molecule, it'll do a couple of rounds like this where it'll go and interact with these non-hepatic tissues that have ABCG1 and the SR beta 1, and it'll keep getting larger, okay? And the LCAT will continue turning that cholesterol into cholesterol esters. And what will now happen is that this HDL molecule in the circulation might come into contact with some VLDLs and LDLs like we talked about here, and it'll use this cholesterol ester transfer protein to transfer some of the cholesterol esters it has to these guys. So like I said, it has more space for cholesterol. But what am I getting at here? Eventually, this guy is going to have to die as well, right? Remember, we said each one has 7 cycles. So what happens to it? What ends up happening is that the liver, okay, the liver also has a scavenger receptor beta 1, but instead of sucking cholesterol out, this one brings cholesterol in. So look at what's going to happen. This guy, this HDL that we had, that mature one, it'll come and interact with the SR beta 1. How does it interact, by the way? It interacts with the help of A1, of course, ApoA1, okay? And you guys know this guy had cholesterol, cholesterol esters, phospholipids, and our favorite enzyme, LCAT, in there, okay? And this SR beta 1 will then bind to this actual A1 right here. It'll internalize the entirety of this HDL molecule, all right? Releasing what? Releasing cholesterol from it, okay? Internalizing and degrading the actual cholesterol, releasing some fatty acids from it, and phospholipids, and et cetera, okay? But that is the actual pathway for how HDL actually circulates, okay? All right, guys, big video. If you stuck around this far, I cannot thank you enough. But that's going to finish everything with this video. If you like it, please comment, please subscribe, and I'll see you in the next video where we start talking about protein overview and protein digestion.